Polycystic ovary syndrome, also called PCOS, is an imbalance of the female sex hormones. The ovaries are part of the female reproductive system, along with the fallopian tubes, uterus, and vagina. If you have polycystic ovary syndrome, your pituitary gland may release abnormally high amounts of luteinizing hormone into your bloodstream, disrupting your normal menstrual cycle. As a result, your follicles do not mature and ovulation does not occur, which can lead to infertility. Some of the immature follicles do not dissolve and remain as fluid-filled sacs or cysts. In addition, your doctor may find your blood has high levels of insulin, a hormone produced by the pancreas. Too much insulin, combined with high levels of luteinizing hormone, can lead to excess production of a male hormone called testosterone in your ovaries. Abnormally high levels of testosterone prevent ovulation, which can lead to infertility.
Regarding the hormonal test, I think the most useful test for diagnosing PCOS would be the testosterone measurement. That means the measurement of your male hormone level inside your blood. Okay. Different hospitals may use different techniques to detect the hormone. Okay. Say some laboratories may use what we call immunoassay. That means an antibody-based kind of the detection method, whereas some hospitals may use the more accurate method, what we call mass spectrometry. For myself, actually, since 2009, I was involved in a study trying to look at the long-term profile of the hormones and also the metabolic parameters in women suffering from this syndrome. We know that according to previous studies, these kind of women may be suffering from a higher risk of metabolic problems like diabetes, lipid problems, high blood pressure, and this may predispose them to have a higher risk of heart troubles in the long term. So we are following up a group of women with this condition, trying to look at what the changes to their blood sugar, their blood lipids, and also the risk on their heart disease in the long term. Now, as I mentioned, for the detection of this syndrome, actually it will depend on several things. First of all, is the symptom, right? The woman has irregular periods, so this can be readily told by taking the history. And for the hormonal test, I think this is readily available in most of the hospitals. And for the ultrasound measurement, again, this is quite readily available in most of the hospitals nowadays. Well, because my research is mainly to look at the long-term problems like the diabetes, lipid problem, and also the heart troubles. So I think this is an important issue because if these problems are detected, then basically we can try to help the woman to do some preventive measures in order to cut down the risk in the long term. So I think this is the reason why we're carrying out this sort of research studies. Well, so far I think there's no effective cure for the syndrome, so to speak, right? And actually the medication that we give is just to help the woman to solve the problem. Say if she doesn't have a proper period, then we try to give them medication to induce the period. If the woman have problem with fertility, then we can give them some fertility drugs to try to induce ovulation. Yeah. So far, the scientists and doctors have not really found out what is the ultimate cause of the disease. Right. So so far we do not have a very effective cure for the problem. But for the problem that are associated with this syndrome, like fertility problem, like irregular periods, then as I mentioned, this can be controlled by the appropriate medication. So this is currently the main aim for the treatment.
Human embryonic stem cell is actually derived from day 5 or day 6 human embryo, which is called blastocyst. And you know, for a blastocyst, it contains the outer layer of a cell type called trophectoderm, which will become the future placenta, while the inner cell clump we call inner cell mass will become the future embryo. And it is believed that these inner cell mass will become any cell type within the human body. So theoretically, these cells, when it is cultured in vitro in a culture dish, when we add in different factors, it can prone to different cell types that we want. So in the scientific field, the researchers are using these cells to derive these cells into any cell type that they want. And these cells can be used as a cell model for disease study or for drug screening. So this is a very important cell at the moment. Human embryonic stem cells are actually like culturing those cancer cells that we need to culture them in a culture dish in which these cells can attach on the bottom of the flask. Then we need to add different a specific culture medium for them to grow in vitro. And then we need to put these culture flasks in a specific incubator, which is like 5% CO2 and it is at 37 degrees Celsius. So these cells can be grown and multiply there in the incubator for 4 to 5 days. And every day we need to change the culture medium. And after 4 to 5 days, we need to expand these cells into a larger flask for them to expand so that we can obtain a large number of cells for our experiments. And if we don't need these cells for immediate experiments, we can freeze them into liquid nitrogen, or we can use them for collect those payload for some gene or protein analysis. So when we need these cells, we can thaw these cells out from the liquid nitrogen and then we can grow them up again. Well, as I mentioned before, human embryonic stem cell have the property to differentiate into any cell type that we want. So after we derive these embryonic stem cell from human embryo, we need to confirm they have this uniqueness. So we need to perform a number of research assays to confirm this identity. If we confirm that these cells cannot be used for further, for research, we have to discard them. So to confirm that the human ES cell, the derived human embryonic stem cell, can be differentiated into any cell type within the human body. Of course, we cannot inject them into the human body to confirm it. We actually have a number of experimental assays to confirm this identity. For example, we can inject these cells back into the immunodeficient mice, and for the teratoma to form. And actually these are the tumor cells which contain different cell type of the human embryonic stem cell. So after a few months of development, we can just dissect these teratoma out and then do some immunostaining to confirm different cell type within that teratoma. So the presence of these cells actually confirm the pluripotency of these human embryonic stem cell. In terms of the duration of the experiment, well, it depends on what type of experiment or what type of result you want to obtain. For example, if you want to do the molecular study of these spheroid, those embryo-like spheroid, we only take like 1 to 2 weeks to finish. However, if you want to know the developmental potential, like what I said before, you want to know whether it can differentiate into any cell type within the animal body, it takes several months to finish. And sometimes we have to repeat the experiments because one time of experimental result is not good enough. We need to do at least 3 to 4 times to confirm the finding is the real finding. So again, it takes longer time to finish. Well, in our department, ONG department, our main goal is to help the infertile couples to have their healthy take-home baby. However, at the moment, there is still no proven predictive tool for assessing the endometrial receptivity. So I believe our currently the newly developed model can help those people and help them to determine whether they are at a good status to have their embryo transfer. And also it helped the clinician in our department as well, because they don't need to repeat a lot of IVF cycle, and that will also help the couples to save their money. This is what we want to help for doing such kind of research.